Simon's doing that, you can, if you've got RStudio up and I wanted to follow along, if you copy what is currently there, there's a setup chunk first, which if you run that means that. Okay, so today we're going to look at how you plot heat maps in R, and then I'm also going to cover some of the kind of principles of scaling data and clustering, really just the basics that you might need to do kind of in preparation for that. So the package that I'm going to be using is the pheatmap package. There are some other packages that you can use, or there's a kind of base R heatmap function which you can use for plotting heat maps. I quite like pheatmap. It has some nice features, particularly with adding annotations to the heat map. So that's the one that I'm going to focus on today. Okay, so if you've got the data that I put up on the chat or from the Google Drive, the first thing we need to do is to read that in. So that needs to be saved in the same location as the R notebook that you're working in. And then I'm just going to assign that to something called. And then if we look at what's that, so this contains names and then normalized recounts for 3 control samples. This is from an RNA-seq experiment, and 3 knockout samples. And then a few extra columns, which we're going to use later to add annotations to the heat map. So for just plotting the base heat map, what you need is to have your gene names, or whatever you want the labels of your rows to be, as your row names, and then just the columns that contain the data that you want to plot. So that will be the 3 control and 3 knockout samples. So I'm going to first select some rows, so that gives, and then convert the names, and that should give us a data frame that has the format that we want for plotting. And then I'm going to assign that to. So if we just put that into the heatmap function, that will plot just our normalized recounts. So you can see there's a load of genes that are not very highly expressed at all. There's some which are kind of intermediate, and there are some which are high. So these are all genes that I've selected as being differentially expressed between the knockout and the control samples. But obviously, if you just use the recounts, you're going to cluster by how highly they're expressed, not by the differences between them. So I'm just going to switch to show some slides to think about what we need to do to this data before we plot it. So this is kind of just a visualization of some made up data where you have some lowly expressed genes where you can't even see that there's a difference between control and knockout, some that are more highly expressed with differences in the fold change between them. So this is kind of what the data looks like in the heat map that we just plotted. So the lowly expressed ones will all be in the blue area, and the highly expressed ones will be in the red area. So the first thing that we want to do is to, for the scale to reflect the fold change rather than the absolute change in the genes. And to do that, log transform the data. So then you can see that actually these 4 genes or whatever they represent have basically the same fold changes as these highly expressed ones, just at a very different expression level. But if you plotted this, you would still end up with all of these being blue and all of these being red. So the next thing we can do to scale it is to subtract the mean or the median, which then brings them all into the same kind of range, and then you're kind of making the most of the color range. Yeah, this would give a much better visualization of the differences between samples. There is a further step that you can take, which is to transform them into a z-score, so where you divide by the standard deviation. And this basically kind of maximizes the difference between your samples. So you do lose some information when you do that, because you can no longer see that these samples had a smaller difference between the control and knockout, or these genes or whatever they are, had a smaller difference compared to these. But if you're just interested in the pattern, then this might be a useful thing to do. Okay, so we'll go back to our studio and then show what those different transformations look like in this heat map. So the first thing we're going to do is to log transform the data. And then if we plot this, so you can now see you're still clustering by the different expression levels. You can sort of see now that there are differences between control and knockout at each of these different expression levels. Okay, so if we now subtract the mean from this, and then plot that in the same way. Now you can see that it's clustering by whether they go up or down in the knockout. So you have this cluster up here, which are genes that are increased in the knockout, and this cluster down here, which are genes that are decreased. Okay, and then finally I'll show the z-score transformation. So we're going to divide by the row standard deviations, but that's the standard deviations from the log transformed, not from the data where we've already subtracted the mean. And if we plot that, so now you can see that this, you can really clearly see the differences between the control and knockout for everything, but you do lose some information. So you can no longer see that the differences for these genes are greater than for the genes down here. And similarly, these genes are also have a higher fold change than these. So you can actually, pheatmap will do the z-score conversion for you. So you could just use the log transform data and include a scale scale by row argument. You can see that basically does the same thing. Something to be aware of if you use any of the other heat map functions is that they don't all do the same thing. For example, the base heat map function will by default scale by row, but it won't look quite the same. So for this, it has to be a matrix. So other than the horrible colors it uses, you can see that the clustering just looks completely wrong. And actually this will it will transform it to z-scores, but it will cluster it before it does that. So that's just something to be aware of if you are using other heat map functions. I think heatmap.2 also, it doesn't scale by default, but if it does scale, it also does the clustering before it does the scaling, which isn't particularly helpful. Okay, so now to talk a little bit about clustering. So pheatmap will by default cluster your rows and your columns. So you can tell it not to cluster. So if you use the cluster cols or cluster rows argument, you can set that to false, and then it won't do any clustering. So in this case, because I've selected genes that I know change between controls and knockouts, actually clustering by column doesn't really tell me anything, and I might prefer just to have the samples put in the order that I give it to them. Yeah, the cluster cols or cluster rows you can set to false, but you can also give it a clustering object that you've generated yourself rather than using the clustering that pheatmap does kind of internally. So the way that you do clustering is first to generate a distance matrix between your genes or whatever it is that you're clustering. So you do that using the dist function, and this will generate basically a matrix which has a distance between every gene, in this case, to every other gene. If you wanted to do it for columns, you would need to transpose this before you run it. Dist will by default use the Euclidean distance between genes or samples or whatever it is. I'm not going to go over the different methods. There are different methods that dist can use, but if you take a look at the dist help pages, you'll see what those are. And Euclidean distance is the distance that pheatmap will use by default as well. So this will have generated exactly the same distance metric that pheatmap will have generated in producing the heat maps above. So once you have the distance measure, you can put that into hclust, which will do the clustering. And then this generates a kind of clustering object. And it tells you that it kind of recognizes that the distances are Euclidean distances, and it uses complete linkage clustering again, there are different options within hclust that can be used, but complete is the method that pheatmap will also use by default. So if we save this into an object, we can then use it in the pheatmap function. So then I'm going to not cluster columns again. And for rows, I'm going to use the clustering object that we just generated. And because we've just used the defaults, which are the same as pheatmap uses, this just looks exactly the same as when you do it just using the default pheatmap. But sometimes it might be useful to have this object so that you kind of have, you have stored the clustering object that was used in here, for example, for extracting the clusters, which I'll show you later. Okay, so sometimes the kind of default clustering doesn't do the best job. One way that you can also cluster data is to look at the correlation between the different genes or samples or whatever it is. Yeah, in that case, it will basically look for things that change with the same pattern, but not necessarily the same magnitude, rather than taking the absolute values. So to do that, you first generate a correlation matrix. So in this case, we need to transpose the data frame before we do it, because otherwise it will the correlation matrix would be between the samples rather than between the genes. So that transposes it. So this will generate a correlation matrix where you have obviously a correlation of 1 between a gene with itself, but then either positive or negative correlations depending whether it's changing in the same direction. So then we can convert that to a distance. For a distance, we want a smaller value to represent something which is to respect genes that are close together. So in this case, we want to use 1 minus the correlation coefficient, because then a high correlation will give us a low distance. Oh, that's right. And by converting it to a distance as well, you can see that rather than be a symmetrical matrix, we've now got this kind of triangular shape. So we've lost the duplication of the values. And then we can cluster that as before. And we won't cluster by columns, and in this case, we'll use correlation for clustering the rows. So you can see that now clusters it in a different way to how it did before. So in this case, it's not really clear whether that's kind of better or worse in terms of clustering. It doesn't necessarily bring out anything particularly new. I just wanted to show one example of where this did really help. So this is an example of a heat map generated where I've clustered based on the correlation, and you can see there are some quite nice things coming out of that. Whereas if the same data was clustered based on the absolute values for my 3 rows, it's just a bit all over the place. So kind of don't necessarily assume that just by running something like pheatmap and using the default clustering, that's always going to do the best job, because it might not. Okay, so pheatmap does actually give you the option to cluster by correlation, so you also don't necessarily need to do the whole calculation, although again it can be helpful to save your clustering object. But you can set, so the clustering distance rows will tell it what distance metric to use. So for the other heat map functions, as far as I'm aware, you can just give it the options that can be used in the dist function, which doesn't include correlation, but pheatmap does include correlation as one of the options there, and then it will do exactly that calculation that we just did. So yeah, that produces exactly the same as above. Okay, so on the right, I've got the help page up for pheatmap, and you can see that there are a lot of options. I'm not going to go through everything now, but I will go through a few of, or some of the kind of main options and ways that you can change kind of the way your heat maps look. Yeah. So the first thing is to look at the color. So this is the default color palette that will be used. So it uses color ramp palette, which generates kind of a sequence of colors ranging between a set of predefined colors. And in this case, it's using one of the Brewer palettes, the red, yellow, blue Brewer palette, and it's generating 100 colors that interpolate between those 7 colors that it's extracted from this palette. But you can put kind of whatever color palette you want in there. So I'm going to use color ramp palette. But I'm just going to give it some colors. So we'll go from kind of a dark blue through white, dark red. Yeah, sorry. And you need to tell it how many colors you wanted to make. So we'll go with 100 again, which is the same as the default does. And then we have kind of blue up to red. So something which you might have noticed on the previous plots, but is even clearer here, is that 0 isn't central in this color scale. Because the values in the heat map go down to a lower absolute value than they go up to, 0 ends up off center. In some ways that doesn't matter that much, because all 0 is representing as a mean and you can still see the differences. But for example, for the SMID2 gene, you can see all the way across it's kind of red. And it is quite nice to have the kind of red versus blue comparison to make the differences really clear. So in order to fix that, you can set the breaks that it uses to assign numbers to colors. And basically what we want to tell it to do is to go from to find the maximum absolute value in the heat map and create breaks that go from minus that value to plus that value. So we'll generate some breaks. So we essentially want a sequence that goes from minus the maximum absolute value to plus the maximum absolute value. And the number of breaks that you want to generate needs to be one more than the number of colors you have, because each subsequent break, any value that lies between two breaks will be assigned to a specific color. So yeah, you need number of colors plus one. So in this case, it's 101. So if we now set our breaks to be these symmetric breaks, then you'll see that 0 is completely centered. Okay, so the next thing we're going to look at is adding annotations to the heat map. So from the original data frame that we imported, we had these 3 columns on the end, which were kind of extra bits of information about our genes. So whether they're a MIC target or not, I've got the log transform mean expression in controls, because obviously once you've done all of the scaling of your data, you no longer have any idea from that of how highly that gene is actually expressed. And then a measure of binding of an RNA binding protein that we're interested in. So pheatmap can plot multiple annotations that appear kind of to the side or above for the rows and columns. And what you need to give it is a data frame or matrix which has row names that correspond to either your the row names of your of what's gone in to make your heat map, or the column names if you're doing column annotations. And then the columns of that data frame or matrix will be the things that you want it to produce kind of an annotation for. So we're going to select out those 3 columns plus the gene names. And then put the gene name to be the row name. Yeah, so then we'll end up with a data frame that looks something like that. And then I'm also going to generate a data frame for to annotate the columns, so that contains the genotype information and then the sex of the mice that we used for the experiment. So In this case, the row names need to be the same as the column names of the matrix or data frame that's used to make the heat map. And I'm going to use grepl to search for control within the column names rather than just type them in and assume that I know what order the columns are in to define the phenotype. Except I need to make that into a factor. And then I'm going to add sex as another column. I think I'm missing and I'll set that to be my column annotation. So now we can add these using the annotation row and annotation col arguments. So I'm going to use the symmetric breaks that we used earlier. So then this produces the same heat map, but it's now added these three annotation kind of columns to for the rows and then two for the columns. Okay, so you can change the colors of these as well. So to do this, you use the annotation colors. And you provide this as a list, and then in that list you can provide colors for as many of these as you want to. You don't have to define all of them, although be aware that when you change one, that might shift what it's using for its default colors for the others. So I'm going to change the colors of the make binding annotation. So when you have a categorical annotation, you need to say kind of what value is mapping to which color. So I'm going to make the non-targets grey. You don't need these in quotes generally, but because there's a hyphen in this, I do. But for the target, I don't. I'll make that turquoise. And then I'll also change the ZFP36 binding one. So because this is a continuous scale, rather than give it kind of a value to a color correspondence, you just need to give it kind of a color palette in the same kind of color palette that you use for the heat map itself. So I'm going to use color ramp palette for that. And do that between white and navy. Oh, and I forgot again to give it a number of colors. So you can see this now changed color, as has this, but some of these have also changed color because it shifts them around when you change one thing. So you could change the colors of everything if you want to. I'm not going to do that now. Okay. So I'm just going to go through some of the other things that you can do to modify how your heat map looks fairly briefly. One thing on here is that the column labels are possibly not the nicest orientation for reading, so you can change the angle of these. There are certain predefined angles that you can use. You can check on the help pages for what those are. So angle col, I'm going to set to 45. And then you can see these have changed around. You can change the font size. So you can either change the font size of everything. So we made the font size 5, then all of the font becomes much smaller. Or you could just do that for rows or columns. So if you do that row, it will change this. As far as I can work out, I don't think there's a way to specifically change the annotation label size other than changing the font size of everything. You can change the color of the border that's around the cells. So you can do something that looks really garish. Yes. Or you can take that out altogether by setting it to NA. So then you don't have any borders. But I think it's probably better with. You can change the heights of your dendrograms. So using tree height of row or column. Tree height of row. And that makes this a lot more kind of squished up. You can remove your legend. You don't want that displayed. So legend will take away the color scale for your heat map itself, or you can use annotation legend, which will remove the legend for the annotation tracks. You can also remove your column or row names. So sometimes you might have hundreds of genes or something, and then you really can't visualize them with these legible, so then you might want to take them off. So then you can say show row names as false, and then they're no longer displayed. You can also you can take away the annotation names as well. So, names row source will remove those. Oh yeah, sorry, except these are the row annotations, so that removes these ones down here rather than these. You can also set your row names or column names to be something different. So if instead of this, we said row, labels row is say 1 to 30, that will then use 1 to 30 as our rows, with 1 being whatever was the first row of our original data frame. Okay, so one other thing you can do is to kind of split your heat map based on the clustering. So you can use this using the cut tree arguments. So we could say split the rows into 4 clusters and the columns into 2. And then this will actually kind of separate out the clusters. So based on the clustering that you've used, it will kind of insert a gap in between the clusters for as many as you specify here. So sometimes that might not be how you want to visualize it, but you might want to have, for example, an annotation bar which shows you which kind of defines your clusters and shows you where the splits are. I mean, in here it's fairly obvious, but when you have many more genes, sometimes it's not really clear to work out where the first few splits are. So to identify the clusters, you can run the cut tree function on the clustering that we generated earlier. So if we do cut tree on the row clusters, sorry, we have to give it the number. That assigns each of your genes a number to show which cluster it belongs to. And then we can put this into our row annotations. Data frame, but because we want this to be treated as a categorical variable rather than a continuous variable, we want to convert it to a factor, because otherwise it will, because it's numerical, it will assume that it's a continuous variable. And then if we run this again, you'll see you now have an extra annotation track that shows which cluster each gene belongs to. So this can be really useful to then have as a record. So if you then say wanted to plot other characteristics of these genes, you then can split them based on their cluster and do whatever you want to with those clusters. One thing that can be useful though then is to have these ordered from kind of 1 to 4 as you go down. So you can see here we start with 4 and then have 1, 2, 3. So you might, after doing this, want to kind of reassign the numbers that they're given. And you can do that by kind of recreating the factor, but reassigning the different labels to them. So in this case, we want to start with 4 and then go 1, 2, 3. But can assign the labels 1 to 4. And then if we plot that again, we can see it's now 1, 2, 3, 4. Okay, so just a couple more things. You can actually kind of extract what pheatmap calculates. So you can assign the pheatmap output into an object. So so you can say silent equals true, which will mean that it doesn't produce a plot. Although you can produce a plot at the same time as doing this if you want to. And then you can assign this into an object. And then yeah, so that then contains these 4 elements, and the first 2 of these are the clustering for your row and columns. So the row clustering basically looks like one of those hclust objects that we generated earlier. And actually, so you could, rather than do the clustering yourself, extract what pheatmap has done and then generate the clusters based on that, which will give you exactly the same output as we got before. And then the final thing that I'm going to show is that you can, pheatmap can send your plots directly into a file if you want to as well. So if you add a file name, there are a few different options for the format that can be, and it will recognize the extension you put on the file name and generate that type of file. If you check on the help pages, it will tell you what the options are for that. But I'm going to generate a PDF. So if you run that, it won't then generate a plot within R, but it will generate a file, which when it opens, I can share. So yeah, that has generated a PDF version, and you can set the height and width of that in the same way you would if you were plotting out to a PDF or something from R generally. Okay, so I think that's everything I wanted to cover, but if anyone has any questions, please either put something on the chat or raise your hand and then I'll try and answer those.